Hey, we're really excited to have Lukas Weber join us and share his research with us. So Lukas comes to us via actually Zurich. So he got his PhD in Zurich and studied with Mark Robinson. So if you guys have ever used done differential expression analysis, you should recognize that name because he's worked on packages such as EdgeR. Then he moved to Johns Hopkins, where he's been working with Stephanie Hicks, a friend of many of us in the group. He's done a lot of really great work just developing computational and statistical approaches to analyze multiomics and high-dimensional data sets. And he's been very successful. He's very well published, so you guys can look at his publication record, but just a few highlights. So we're very proud to have him recognized as an early career symbiont. If you guys are familiar with that award, it's an award for open science and was developed, actually started by Casey back in the days and was awarded at Pacific Symposium for Biocomputing. And he also was a recipient of the K99R00 mechanism, where he's looking to, and I'm sure you'll probably talk a little bit about this today, is looking at tools to unsupervised methodologies for analysis of kind of spatially resolved data. So I think with that, I will hand it over to Lukas. Thanks for joining us today, and we're excited to hear about your research.
Thank you very much for the nice introduction. I'll share my slides. Now, is that full screen? That's good. Okay, great. Okay, thank you. Yeah, so my talk will be about unsupervised statistical method development and data-driven analysis workflows for spatially resolved transcriptomics. And yeah, thanks. We just had some quick background about me. So I'm an applied biostatistician working on methodological development and collaborative analyses for analyzing high-throughput genomic data in general, and with a special focus now on spatially resolved transcriptomics and single-cell data. I'm currently a postdoc at Johns Hopkins in the Department of Biostatistics, and previously did my grad studies at the University of Zurich in Switzerland and ETH Zurich in Switzerland. So I've structured my talk around a series of research themes, and here is an overview of all of this. In the middle, so my overall theme throughout my research has been on methodological development and collaborative analyses for high-throughput genomic data. And I've worked on several technological platforms, or data from several technological platforms over time. And that includes high-dimensional cytometry, which was during my PhD, and then spatially resolved transcriptomics and single-cell and single-nucleus RNA sequencing more recently. And then at the top left there, that leads into research theme 1, which is on unsupervised statistical methods development in these areas. And I've listed there 2 papers in this theme. The first one there on spatially resolved transcriptomics methods. I'll talk in detail, so I've highlighted that with number 1. I'll talk about in detail about that paper in the following section on a new method called nnSVG for identifying spatially variable genes in spatially resolved transcriptomics data. And that's currently available from bioRxiv and is also in revision at Nature Communications. And earlier there, the other paper that's in the context of high-dimensional cytometry. And then in the second part of the talk, I'll talk about research theme 2, which is on collaborative analyses. And I've done collaborative analyses in several contexts. Most recently, I've focused on neuroscience, and I'll talk in detail in the second part of the talk, the number 2 there, about a recent paper where I'm co-first author on analyzing single nucleus and spatially resolved transcriptomics data from a region in the brain called the locus coeruleus. And that's available for bioRxiv and also in revision at eLife at the moment. And I've previously also done several collaborations in context of cancer and immunology. And then research theme 3 at the bottom, this touches on all of my other research, and I'll talk about this more briefly towards the end. I've been involved in a number of projects on benchmarking analyses, where we try to compare different methods to establish which methods work well under which circumstances. I've done that in the context of cancer and also a review paper there, and several independent benchmarking works. And also I'll talk about analysis workflows and tools, where I've been involved in the R and bioconductor communities for some time, and open source software development and reproducible research. And at the end, I'll talk briefly also about my K99 R00 award on the right there, which is on unsupervised statistical methods development and data-driven analyses for spatially resolved transcriptomics. Okay, so then for that first paper that I'll discuss, I'll start with some background on spatially resolved transcriptomics. So this refers to a class of new technological platforms that allow us to measure transcriptome-wide gene expression at spatial resolution in tissue samples. And as an example here, I've got some schematics from the 10x Genomics Visium platform, which was released in 2019, and which is currently the most widely used platform for this type of work. The way this works is you have these tissue samples, tissue slices, and then a grid of spatial locations on a slide where mRNAs are captured at each of those spatial locations, spatially barcoded, and then sequenced. And that allows us to recover the spatial locations from where those measurements came from. Then as an example there in the middle, then we can look at the expression of individual genes across the full transcriptome at each of those points. And the dimensions here in this particular platform, so each of these tissue slices, it's 6.5 by 6.5 millimeters in dimensions, and approximately 5,000 of these spatial locations. And on the right there, a schematic from the 10x Genomics website about how the capture and sequencing works. And as a further illustration here, putting this into the context of bulk RNA sequencing and single cell RNA sequencing. So spatially resolved transcriptomics can be compared to single cell RNA sequencing, but now we also know where the measurements came from in the spatial context, although depending on the platform, it's not at exact single cell resolution. So in this example, we have different cell populations within the brain, where the Lego blocks represent cells and the colors represent cell populations. In bulk RNA sequencing, they're all mixed up in one sample. In single cell RNA sequencing, we can resolve the individual populations, but we don't know where exactly they came from. And then in spatially resolved transcriptomics, we also have that spatial information. Oops. And the types of analyses that we're interested in here, I'm especially focused on unsupervised and discovery-based analyses, such as feature selection, where we identify spatially variable genes, clustering to identify spatial domains or spatially distributed cell populations, and differential gene expression. And as a few examples here on the right from a few different papers, the top one there, that's showing an example of individual genes with spatial patterns of expression that we refer to as spatially variable genes. That's in the mouse olfactory bulb example there. Then in the middle, that's an example from cortical samples in human brain, where we're interested in the cortical layers, which have this layer structure there. So trying to identify those spatial domains. And the example at the bottom is from tumor samples, where we have different spatial domains representing different tissue types. And we're interested in characterizing cell type and state heterogeneity throughout this, and also identifying biologically informative marker genes for individual structures or anatomical features. And some more information here about the data structure. Here I'm comparing it again to single cell or single nucleus RNA sequencing, which have been around longer. So in both cases, we end up with tables of expression counts, gene expression counts, usually arranged as genes by cells in the case of single cell data. In spatially resolved transcriptomics using spot-based platforms, is genes by spatial locations. But then in addition to that, we have the spatial coordinates for each of those measurement locations. And further, it's also possible to extract image features from the histology images, such as number of cells per spot or cell size and morphology. And then spatially variable genes, this is the core concept in the methodological project that we've done. So here I have an example to show what we mean with this. This is from dorsolateral prefrontal cortex samples in post-mortem human brain samples that were measured with the 10x Genomics Visium platform. This was a collaboration that I was involved in 2019 and 20, published there in that paper, where I'm a co-author. My role was on doing unsupervised analyses, developing an unsupervised analysis workflow. And we've used this data set for our further methodological work now as well. This example here on the right is showing expression of 6 different genes that have very strong spatial patterns. The top 3, MOBP, PCP4, SNAP25, are associated with the cortical layers or with the gray matter versus white matter distinction. And the bottom 3, HBB, IGKC, NPY, those are associated with blood and immune processes within that same sample. So this is showing 6 times the exact same sample and showing expression of 6 different genes. And the really important part here methodologically is that these different genes have very different spatial ranges in their expression patterns. And we want a method that can identify all of these in an unsupervised manner, regardless of the different sized pattern. And also some externally sourced ground truth labels there at the bottom, which we use in the evaluations. So the bottom left, that's showing the 6 cortical layers and white matter, where white matter is black. And the bottom right is showing white matter versus all the layers, gray matter. So we can see the top 3 there are clearly associated with those cortical layers, and the bottom 3 are not. And we want a method that can identify these genes in an unsupervised manner and give us the top genes that are most strongly associated with the biological features in this data set. Okay. Then that brings me to that first methodological paper on a method called nnSVG to identify and rank these spatially variable genes that we're interested in. So the motivation here was, so why are we interested in these spatially variable genes? This fits within a typical analysis workflow for this kind of data. I've got a schematic here. This fits within the feature selection step, which is part of pre-processing, where we try to find the most informative genes and then use that for further downstream work, such as clustering to identify spatial domains. We use this for two purposes, really. First, also just data pre-processing to reduce the number of genes to a smaller, more informative set, which reduces noise, improves computational performance, such as top 1,000 informative genes. And secondly, also identifying the top ranked genes for further. Someone's got a phone going. Okay. And secondly, also identifying top ranked genes for individual further analysis or validation. And the example there again on the right, those 6 top genes that we are particularly interested in from previous work in that data set. So then the issue here, when we were doing this work in the collaborative analysis at that time, was that existing methods to identify these spatially variable genes either were far too slow or did not perform very well for various reasons. And that motivated this methodological project. I've listed here 2 baseline methods that we can use to identify informative genes, one called highly variable genes, which does not take into account any spatial information. That's sort of the standard method from single cell analysis workflows. And secondly, there's also a measure in the statistical literature called Moran's I statistic, which quantifies spatial autocorrelation. But both of those are quite simple and not adapted to this specific data type. So then several new methods were developed by other groups at the time when these data types were first coming out. And that first one I've shown there, spatial DE, that was quite a prominent method developed by Svensson et al. This used Gaussian process regression and the likelihood ratio test to identify, to rank and identify all spatially variable genes in a data set. And that's a very nice method, but there was the implementation scales cubically in the number of spatial locations, which was fine at that time in 2018. The platforms at that time only had a few hundred spatial locations. But then when we were applying these methods in the newer Visium data, with thousands of spatial locations, and now even more in future platforms, that becomes completely infeasible with the cubically scaling method. And secondly, there I've also shown SparkX. That was a newer method that had a very nice statistical method to make this much faster, but that method lost sensitivity to identify spatially variable genes with different types of patterns within the one sample, which is what I've highlighted there with those red arrows. So having these genes with really large spatial ranges and really small spatial ranges within the same data set, we want to be able to recover all of those without making strong assumptions. And there are the model formulas from the Spatially method, which we were inspired by. So it assumes log transformed normally distributed data and then an assumed kernel or covariance function. They use a squared exponential there. That L parameter that I've highlighted with the red arrow, that quantifies those ranges in the spatial expression. So we wanted to retain that sensitivity, which SparkX does not have. So then we worked with our collaborators here at Johns Hopkins in the Biostatistics department, Dr. Abi Datta there, the first author of this paper. They had previously developed a statistical approach called nearest neighbor Gaussian processes, which uses an approximate likelihood with a small set of nearest neighbors, like 10 to 15 nearest neighbors out of these thousands of spatial locations that we have. That approximates the data very, very well. Which they, in that paper that I've shown there, they show that it retains almost all of the information. And crucially for us, it gives us linear scalability in the number of spatial locations. And that's because we have a sparse precision matrix that can be inverted much more easily than the full covariance matrix. So we combined this with the likelihood ratio test approach from Spatially to implement a new method that we called nnSVG. So we use a package called BRISC there that was implemented by the authors of the NNGP paper previously for the individual model fitting. And we fit one model per gene and extract maximum likelihood parameter estimates and log likelihoods and compare against a model without any spatial terms. So on the right there, I've pasted in the model formulas again. And so we fit this individually per gene and compare against a model without any spatial terms and calculate a likelihood ratio statistic per gene. That likelihood ratio statistic lets us rank all genes by effectively the strength of the spatial patterns per gene within the data set. And really importantly, we keep that gene-specific sensitivity. So we have that gene-specific length scale parameter, L, that I've highlighted again with the red arrow. Although we use an exponential covariance function, not a squared exponential covariance function, there was some evidence in another paper showing that the squared exponential drops off too fast with distance in this kind of data. And optionally, we can also include covariates for spatial domains in the design matrix X. And what that means is then we are looking for spatially variable genes that have a pattern of expression within a subregion of the tissue slide, not within the whole region. Yeah, and then the outcome of all that is that we have a ranking of all the genes in the data set by the strength of their spatial patterns, respectively. We can also calculate an effect size there at the bottom right, which is the proportion of spatial variance out of all variance per gene. Okay. So then in the next few slides, I have several evaluations where we tried this out on several previously analyzed data sets to check whether we recover known signals that we know are in these data sets. So this is that same dorsolateral prefrontal cortex dataset from 10x Visium platform that I've mentioned previously. And here we know from the previous work that these 6 genes especially are very, very highly informative. So we want to make sure those come out as highly ranked. And then we also have a longer list in our paper. And one evaluation here, this is, so first we check, do these gene-specific length scale parameters that I mentioned a few times, do the estimated parameters make sense? So here I'm showing a density plot across all the genes with the estimated length scale per gene, and showing that for the three at the bottom on the left there, with the small patterns, they do indeed have very small estimated length scale parameters, which is good. And the three at the top with much larger ones have larger parameter estimates. So that gives some confidence that the parameter estimates are sensible. And then in this part of the evaluations, I'm taking those 6 genes and calculating the rank of them within the full list of spatially variable genes in this data set. So here I've got 6 genes on the horizontal and the rank on the vertical. So we want to be as far down as possible. And I'm comparing here 4 different methods: nnSVG, which is our method in dark blue; SparkX, which is the competing one that's also fast; and then the 2 baseline methods, HVG is the non-spatial baseline, and Moran's I spatial baseline. And we can see that nnSVG identifies all 6 of these within the top 300 or so genes, which is great. HVGs actually works even better on this data set for the small length scale ones, those HVB, IGKC, and EY. And then in our paper, we have several different data sets. In some of the other data sets, HVGs does not work well at all. So we have the best balance across the different evaluations. And in this data set, SparkX doesn't work at all for those small length scale genes. It doesn't rank them at all within the top 1,000 spatially variable genes. And that's because it doesn't have that sensitivity to the different length scales across genes. So that's the overall ranking of methods for this data set. And then we do several further evaluations in our paper. Here, we also have one quick check at the end, just checking the expression patterns of the highly ranked genes. Do they look sensible? We know in this data set that the strongest signal is between the gray matter and the white matter, which is the bottom left region versus the rest. And this is showing the top 20 spatially variable genes ranked in order from nnSVG. And most of those are very clearly associated with that white matter versus gray matter signal, which is good. So then I mentioned we had several different data sets. This is now another data set from mouse hippocampus using SlideSeq platform, which is a different spatial platform. And here we're interested in identifying spatially variable genes within a subregion of the tissue area, not within the whole tissue area. On the right, that's showing the whole area with annotations for cell types. And we're interested in identifying spatially variable genes within that pink region, which is the CA3 region of the hippocampus. And in the previous work on this data set where they published this, they showed that there were several genes, and more than just the 2 I'm showing here on the left, which have very strong gradients of expression within that CA3 region of the hippocampus. So we want to check that those come out as highly ranked. So those 2 example genes on the left there. And here I'm doing the same type of analysis with ranks per gene. We're showing those 2. And we see here in this data set, both nnSVG and SparkX identify both of those within the top 300 or so genes, which is great. But now HVGs here does not work at all. It doesn't identify those within the top 1,000 genes. And then we have a couple more data sets in our paper showing that we get good performance across a range of settings. And this, and so this is with the covariates setup. Yeah. Okay. Then we also do some simulations to check that we really do get this linear scalability, which was really the underlying motivation for this project, that we just did not have a method that retains sensitivity and is fast at that time. But we really do achieve linear scalability, which is great. So here I'm subsampling a number of spatial locations for each data set, and then running the method a number of times for one gene, and confirming that as we increase the number of spots on the horizontal axis, then runtime scales linearly on the vertical axis. So in summary for this part, we developed a new method to identify spatially variable genes, which we show in several data sets outperforms existing methods and baseline methods. and identifies known spatially variable genes in several previously analyzed data sets. And we have this sensitivity due to the flexible length scale parameter to identify genes from different size processes within the one sample, which a competing method cannot. And linearly scalable in the number of spatial locations. So this can scale to large data sets. And preprint is available from bioRxiv. And then some words about the implementation and reproducibility. So we have an R package implemented within the bioconductor framework for running this method with lots of documentation and tutorial, and also a GitHub repository to reproduce all the analyses and figures. And the data sets are available through a separate data package as well. Okay. Then that brings me to research theme 2, which is on the collaborative analyses. And here I'll discuss a recent collaborative analysis of a data set from postmortem human brain samples, which is that co-first author paper I've listed there with the number 2. So the background here is that we're interested in, so on the right there with the red circle, in this schematic of the brain, I've highlighted the locus coeruleus, which is a small cluster of neurons within the brain stem. It's a very small number of neurons, but they're very, very important. So these are norepinephrine producing neurons. They project widely throughout the whole brain and central nervous system. They have critical roles in a number of brain functions and have been implicated in neurodegenerative disorders, especially Alzheimer's and Parkinson's, and also degeneration with age. And this region has been relatively understudied in humans due to its small size and inaccessibility. And having these spatial platforms now presented a wonderful opportunity to try to characterize the transcriptome-wide expression landscape of this region in much more detail than was previously possible. So the aim of this study was to characterize the transcriptome-wide gene expression landscape of the human locus coeruleus at spatial and single nucleus resolution, which hadn't been done previously in human samples. And here I have an overview of the experimental design. So this figure on the right is created by my co-first author, Ina Devicha from the Lieber Institute for Brain Development. So we have 5 neurotypical adult human brain donors. And then we have spatially resolved transcriptomics data from the 10x Genomics Visium platform and single nucleus RNA sequencing data. In the Visium data, it's 8 samples from 4 of those donors that ended up passing quality control. And in the single nucleus data, we have a bit over 20,000 nuclei from 3 donors in the end. And we identify those regions. So in the top left on the schematic on the right, that's actually showing the tissue block and where this is located within the brain stem. So the locus coeruleus can be identified by pigmentation and cell size. It has this distinctive dark blue color. So we can identify that region within the Visium samples based on the histology images and then do our computational analyses within that correct region. And at the top right, that's confirming expression of several known marker genes with single molecule fluorescence in situ hybridization with RNA-scope, confirming that we have individual neurons from that population we're interested in. And at the bottom right, showing schematic of the single nucleus and spatial workflows. My role here was in developing an unsupervised discovery-driven analysis workflow to characterize the gene expression landscape in these samples. So one thing we did was differential gene expression testing in both the Visium and the single nucleus data. Here in the Visium data, we compare manually annotated regions per sample, where we identify based on the pigmentation and cell size, we identify that that's the region where the locus coeruleus is located. I didn't mention, so the locus coeruleus, the size, it's about 2 by 2 millimeters if it's a section like that. So that fits onto the whole Visium slide. So we've got these manually annotated regions, then we do differential gene expression testing between those regions and the rest of the sample. And here I'm showing that we recover several known norepinephrine NE neuron marker genes, and then in addition to that, generate a long list of additional unsupervised genes associated with these regions. So this heat map is showing expression in the LC, locus coeruleus regions on the left, and in the non-LC regions. And I'm showing the top most highly ranked genes from the differential expression testing. The ones I've highlighted in red are well-known marker genes from previous work in mice. So we confirm that we have those. And then, yeah, in total, we get 437 genes in our list that we report. Here's also in a different representation using a volcano plot where we compare significance against the effect size fold change. So we want the ones that are the sort of very clearly at the very top right there, and also in general towards the top right or top left. That was in the Visium data. Then in the single nucleus data, here we did an unsupervised clustering workflow to see if we could recover that population of NE neurons, norepinephrine producing neurons, which have never really been characterized at the single nucleus level in humans. And we were able to successfully do that. So this plot is showing the result of that clustering. Here I'm showing along the horizontal axis at the bottom, that's showing known marker genes for several different neuronal and non-neuronal populations. And in the rows, in the annotation on the right, that's showing 30 clusters that I got from this workflow in the end. And then I've grouped them into different neuronal and non-neuronal populations based on those known markers. And that one that I've highlighted there in red, that's a very clear cluster of NE neurons with very high expression of those 4 known marker genes there, which was, yeah, was really an amazing result to get this very clearly at single nucleus resolution. We have this very clear population. And so then we've identified it here based on these few marker genes, and then we have the full transcriptome-wide expression profile of that same population that we can report. It was very small, so only 295 nuclei in the end, which was not very many. And at the bottom left there, also a UMAP representation of the same thing. Then we also did differential gene expression testing between that neuronal population and all other neuronal populations to further characterize the expression landscape there. And similar to before, we got a long list of genes associated with this, but now at the single nucleus resolution instead of the aggregated spatial resolution in the other data. Here I'm again showing expression within that NE neural population and all other neuronal populations. And I'm highlighting in red a few known marker genes that we use as checks that we have those, and then we have the full list of additional genes for results. And then in addition to that, there were several unexpected results, which were very interesting here. And this one was very nice for me. So we also applied the nnSVG method that I introduced at the start. We applied that as an alternative method to identify genes associated with the locus coeruleus regions in the Visium samples as spatially variable genes now, instead of using differential testing, and then we can compare those. And one thing that stood out here was that there were several genes associated with, or that were known to be associated with different region in that brain, different structure in that brain region called the choroid plexus, which we were not expecting. And these results from nnSVG allowed us to investigate this and figure it out in more detail. So what I'm showing here is the 8 Visium samples that passed quality control. The red regions in each one of them are the regions containing the locus coeruleus. And then I applied nnSVG to identify any gene that has a strong spatial pattern in an unsupervised manner across the whole slides. And our list of top genes from nnSVG included some unexpected choroid plexus genes. And then by going back here, we saw that those genes were just in that one sample, which is the one where I've also got the histology image at the bottom left. So we see a streak of expression there of just a small number of genes within the sample from that one donor. So that was where we had this little slice of choreoplexus within the samples, which in a way is contaminating our results from the locus coeruleus. And we were able to find this because those genes came out as very highly ranked spatially variable genes when we applied nnSVG. And that makes sense if we look at that pattern there. There's a very strong spatial pattern there in those two streaks. And so by identifying that lets us exclude them from our final list of locus coeruleus genes. So that was a really nice result to apply this method nnSVG to find something here that we would have missed if we had only had a more standard analysis with the differential testing. And then a second unexpected result that we saw was that we had a very high proportion of mitochondrial reads in some nuclei and in our single nucleus data. And so that didn't make a lot of sense. We don't expect mitochondrial reads in nuclei, but they're not supposed to be there. And usually we actually use mitochondrial proportion as a filtering threshold in our quality control steps during these pipelines. But what happened here was that there was a combination of both biological effects and technical effects, which meant that we had high mitochondrial proportion in exactly that population of NE neurons that we're interested in. This was from the biological perspective. This is because these neurons have very high metabolic demand due to these long-range projections that go throughout the whole central nervous system. So very high energy demand, so elevated expression of these mitochondrial genes for that reason. And then at the same time, some technical effects where some fraction of those mitochondrial reads are captured within the droplets in the single nucleus data. And this ends up being crucial during the pre-processing analysis. So if we just do a standard filtering threshold of say 10% mitochondrial is a bad measurement. Beyond that, then we would have filtered out this whole population that we're actually mainly interested in, which is the NE neurons. In this plot here, I'm showing the 30 clusters in the single nucleus data, and then mitochondrial percentage per nucleus. And we see that cluster of NE neurons, which from all of our other results, which I haven't shown everything here, we're confident that that is good data, but we do have a high mitochondrial proportion in that one population. So this showed us that we really need to adapt our pre-processing and other workflows in this type of analysis based on the specific context of the data that we're analyzing. This was very interesting. Okay, so in summary, this was the first characterization of the transcriptome-wide gene expression landscape in the human locus coeruleus at both spatial and single nucleus resolution, and several additional results that I didn't cover, and the preprint is available and it's in revision. And as previously, we made all of our code and data available. So we have a bioconductor package with downloadable R objects in that format, and then also web apps to explore the data interactively through a browser. The top one there is for BCM data and the bottom one for the single nucleus data. And a GitHub repository to reproduce all of our analyses and figures. So in the last few minutes, this brings me to research theme 3 on benchmarking, analysis workflows and tools, and open source software and reproducible research. So I've been involved in a number of benchmarking studies over the years. And in these studies, we're interested in identifying which methods perform well for which types of data or which types of analyses. And in this example here, with co-authors from here as well, so this was published in 2021, we were interested in finding out whether genetic demultiplexing algorithms. So these are algorithms that, so we want to pool our single cell RNA sequencing data prior to library preparation from multiple donors, which can reduce costs and batch effects. And then we can computationally demultiplex those based on variance per donor, if these are from distinct donors. So several algorithms exist for that task, but we didn't know at the time whether these algorithms still work well in the context of cancer, where additional somatic mutations might interfere with the variant signal per donor. So we tested this on data sets from high-grade serous ovarian cancer and lung adenocarcinoma and showed that it still works well. And this is an example of the type of analysis that we did here. So several simulations here with a certain proportion of doublets and ambient RNA, and then evaluating precision and recall in assigning cells to their correct donors per sample for various combinations of the methods. So that's just as one example of this type of work. And I've also previously worked on a guidelines and review paper where we tried to provide advice on how best to design a benchmarking study, and especially to distinguish between independent benchmarking studies where we are not involved with the development of any of the methods versus benchmarking during our own methodological development of our own methods. And I've done studies of both types. On the left, are independent benchmarking studies that I've previously done, and on the right, during my own methodological development. And analysis workflows and tools. So I've recently, so this is a co-first author paper, we've developed a data structure for storing spatially resolved transcriptomics data sets within the bioconductor framework in R, so storing things like spatial coordinates in specific slots. And that can be used for integrating within longer analysis workflows. Previously, also, I've worked on analysis workflows in the context of high-dimensional cytometry. So this paper here, perhaps a contributing author, showing a long example workflow for how to do analysis workflows in high-dimensional cytometry. And some comments here about reproducibility and open science. So I've always tried very hard to make sure all the methods I develop are freely accessible as open source software packages. So some examples at the top there, nnSVG and Diffsite within Bioconductor. And then always making code available to reproduce analyses, for example, through GitHub repos, and making sure all of our data is available in the form of data packages or managed repositories or other sources, and posting preprints. And as was mentioned at the beginning, so yeah, I just want to mention that I received this nice Research Symbiont Award in 2021 in recognition of my efforts for code and data sharing. Okay, and in the last couple of minutes, that brings me to the K99 R00 award. So I've been awarded a K99 R00 grant from NHGRI on unsupervised statistical methods development and data-driven analysis for spatially resolved transcriptomics. And I'll talk about this in some more detail as well tomorrow in my chalk talk. So here, just a quick overview of the aims, aim 1, aim 2, aim 3. So aim 1 is about methods for pre-processing and feature selection, where one of those sub-aims is nnSVG on spatially variable genes that I discussed at the start. And then aim 2, building on that for downstream methods such as spatially resolved clustering. And aim 3 on data infrastructure and benchmarking resources such as the spatial experiment structure that I mentioned. And I think I'm almost out of time, so I'll leave this one, but I'll want to briefly mention one of those, aim 3B there, that's on developing an online resource of analysis workflows for spatially resolved transcriptomics. So that's a work in progress. I think this will be really helpful for showing people how to connect these different methods into full analysis workflows. So this is an online book down resource where we have interactive examples with R code and data sets showing how to step through analyses. And yeah, this will be extended. There's currently a draft version available in my website. Orchestrating spatially resolved transcriptomics analysis with bioconductor. And my long-term plans will be to continue in this direction of statistical and machine learning methodological development, really driven by biological collaborations in areas including neuroscience and cancer, and with a focus on single cell and spatially resolved transcriptomics. And I will be motivated by the development of new platforms as these are developed over the years. And I have experience in applying for funding, currently funded by the K99-00. I've also applied for some additional funding from Chan Zuckerberg Initiative recently. And I have previously applied to the Swiss National Science Foundation during my PhD. And I will plan to apply for R01 funding during my initial years as tenure track faculty. And with that, I'd like to thank especially my two primary advisors on the left, postdoc advisor, Dr. Stephanie Hicks at Johns Hopkins, and my PhD advisor, Mark Robinson at the University of Zurich in Switzerland. And on the right, all of my main collaborators from the projects that I've presented. And that leaves some time for questions.
Hi, I'm Nick. My question is, it seems like an obvious thing, but maybe because I have no idea what I'm doing, to take little tiny slices and put them on your imager one after the other and create a 3D data set. Have you thought of that?
So you're asking about integrating, so in this, like in the so with the Visium data, integrating multiple samples into like a single 3D sample. Yeah, so I have seen data sets where the people are basically taking serial sections across a tissue sample, but really fully reconstructing that into a 3D sample. That's very challenging. I think this field is going in the direction of data sets with much more samples essentially, and then that sort of work will become more feasible. But I think that's still at quite an early point at this stage.
Another quick question, just to make sure I understand. Each of those little circles in your Visium data set, does each circle provide you an entire sequence?
Yes. So each spot, you mean? Yeah. I'll go back to that schematic at the beginning. Yes. So each of those spatial measurement locations, like in the there. So you're measuring transcriptome-wide gene expression at each of those spots. So it's the dimensions is about 5,000 of these spots in the Visium platforms across a tissue area of 6.5 by 6.5 millimeters. And you're measuring transcriptome-wide gene expression at each of those spots, which is kind of analogous to single cell data, except you might have more than one cell per spot, depending on the tissue density. In human brain, it's about 1 to 10 cells per spot.
Thank you.
There's some in the chat. I can have a look. Thanks. Yeah, the link.
So Lukas, great talk.
Thank you.
I love the way biostatisticians just take things apart. It's it's really fun to watch it. Just rip every little parameter down to shreds. I'm curious, since you have this nice linearly scalable and tunable really parameter for length scale, I'm wondering whether or not you have any thoughts about sort of how to automate the discovery of what that length scale parameter should be, potentially within one whole slide, but also potentially like you're showing here within regions to try to maximally capitalize on the variability.
Yeah. So here we've tried to do this in a way where we have minimal assumptions. So we fit this length scale parameter individually per gene, and that lets us have this sensitivity where in the final ranking after our likelihood ratio tests against models without spatial terms, in that final ranking, we get both genes that have, say, really large spatial range and ones that have really small spatial range, which in this data set happens to happen because we have these large cortical layers and at the same time have these small structures from blood and immune processes, which was a little unexpected. So we tried to make as few assumptions as possible and to really have that individually per gene, because as soon as we start pooling this information across genes, then we would lose that sensitivity. Like if 90% of our genes here are associated with the cortical layers and only a small number associated with the blood and immune processes, that would kind of wash out while when we do that averaging. Yeah.
Sorry, I missed the detail that you were fitting this on a per gene basis. That's awesome.
Yes, we're fitting this on a per gene basis. That was really the innovation in this method, that we were able to do that fast enough with the linear scalability to really fit that individually per gene. Yeah, and in fact, that one competing method, SparkX, that does pool the information across genes, and that's exactly why they lose that sensitivity. So they get really excellent computational performance and speed for that, but then they lose that sensitivity to the different biological processes within the one sample. Yeah.
So kind of building off of Sean's line of questioning there, I mean, have you looked at doing the analysis with short and long separately or medium scale? Because you can potentially get after some of those questions and they're going to give you different results.
Yep.
If you're going to use genes that have longer scales and genes that have shorter scales, and they could get after different aspects of the biology.
Yeah, I think that would make a lot of sense in a data set like this, where we already kind of know what we're looking for. Like here, we know that we have these large ones and the small ones, but for general application of this method, our intention is that this can be done in a relatively exploratory and unsupervised manner where we don't really know what the structures are going to look like when we start the analysis. So by making those kinds of assumptions in advance, it's kind of circular then. Like we can only make those assumptions if we already sort of know what we're looking for, which in most cases we don't when we're applying these kinds of methods. So we've really tried to implement this in a minimal assumption manner where it's just like in the locus coeruleus example that I mentioned there, I just run nnSVG across the whole sample without making any assumption about the shape or the size of the region that we're interested in. And that gave us really good results and makes it easy to apply as a first analysis during these discovery-driven workflows.
Yeah. Yeah, I mean, I wonder if you could provide some type of guidelines, as you were kind of alluding to at the end of your talk, about the types of questions and the types of biology that can be revealed by the different length scales. So, I mean, in cancer, I'm very interested in understanding how local immune invasion affects tumor growth and response, et cetera, et cetera. And there you might want short length scales versus maybe longer. Maybe the brain might be a place for longer length scales and things like that. So yeah, anyway.
Yeah, that would make sense. Yeah, depending on the exact tissue type that you've got. Yeah. And also then within the sample, like if you have like a large region of tumor cells or something, then you have maybe some genes associated with that. But then in the other part, you might have some immune infiltration on a much smaller scale. So that's really during the interpretation phase. For interpreting those length scales. Yeah.
Great, thank you.
Did I understand correctly? You're only using the the transcriptome data. You're not using any sort of processing of the images themselves?
In this work, that's correct. Yes. So we're using the gene expression measurements and the spatial locations. We're not including any information here from the histology images. Yeah, and I think integrating those will be very promising, I think, for future projects. The simplest information we have is the number of cells per spot, but then also going further and having morphological features, I think, could also really help. Yeah.
Is that part of your RU, or is that kind of subsequent to RU? Or maybe we'll talk about this tomorrow. The R00.
Oh, right. That's not in there. Yes, no, that will be for future work. Yep.
So Lucas, how would you? Is there? Does the estimates that you get on a per gene basis? Are those informative for the purposes of downstream differential expression analysis? Are there any parameters that you gather that could be thought of as sort of weights or variances? Yeah.
No, so for differential expression testing, where we're comparing different regions, so there I could imagine using those the number of cells per spot. Actually, that would be possibly an informative weight parameter there. Yeah, that could be used. But here from these length scales, no, because that's at the gene level, not at the spot level. Yeah.
Thanks. I guess one more question on these lines. Is it, when you're fitting these things, is there, so you mentioned about not pooling, but pooling is a little bit different than using, for example, methods like ND-seq, where you're taking variants and you're borrowing information across regions or cells or genes.
Yeah.
I'm just curious if you have thought about that, and if maybe it's in here already. I don't know. I'm just curious.
No, it's not in here yet, but I could imagine concepts like that being useful where we have multiple samples. So if we have the same gene across multiple samples, could we then improve power by having some of those ideas, that the borrowing of information across samples instead of genes? I think that could be promising here.
Yeah, this is a shameless promotion here, but there's going to be a seminar next by Sky Lee, who's the manager of the Genomics Shared Resource, who's going to be talking about the technology to generate spatial transcriptomics in single cell. So if you guys are interested in that and have the next hour free, please join that seminar. So.
Oh yeah, and that's in the chat. So
yes, I put that in the chat. Yes.
All right, everyone. Thank you so much, Dr. Weber. It was a great presentation. We hope everybody can also attend tomorrow's Chalk Talk. We can ask more questions. But thank you, everyone. Have a great afternoon.
thank you.